Now let us study about the second event in the sexual reproduction in plants, that is fertilization. In flowering plants, the fertilization process that occurs is known as double fertilization. As stated, after the pre-fertilization process, that is after the pollination that occurs, the pollen grain gets attached to the stigma of the flower, whether it may be self-pollination or cross-pollination. After getting attached to the stigma of the flower, through pollination, pollen tube gets formed. This pollen tube contains two male gametes which enters into the ovule, which is also known as an embryo sac. Now, as stated earlier, within an embryo sac, there occurs a megaspore mother cell which undergoes meiotic division and forms four haploid megaspores. Out of which three gets degenerated and only one remains active. This one, that is active one, participates in the double fertilization process. Let us study the double fertilization process that takes place within this embryo sac. The one megaspore that is active produces 8-celled structure. This we have already studied earlier. This 8-celled structure consists of 3 antipodal cells, 3 micropolar cells, that is micropilar cells, and 2 secondary cells. As it can be seen from the figure, this figure depicts the structure of an embryo sac that contains 3 antipodal cells, which is exactly opposite to the embryo sac opening, that is the micropyle. These 3 cells are known as micropilar cells, out of which there occurs 2 synergid cells and 1 egg cell. The 2 nucleus or the 2-celled structure which is located in between is known as secondary nucleus or a polar nuclei. Thus, the pollen tube that is developed gets entered into the embryo sac through the micropilar opening. As said earlier, these pollen tubes contain two male gametes. So it is these two male gametes that enters within the embryo sac. Out of these two, one male gamete interacts with the synergid cell. And the egg cell. This interaction of the male gamete and egg cell is known as embryo, that is a diploid zygote, which is a two-celled structure. This we'll study later in the post-fertilization events, which is embryo formed due to combination of male gamete and egg cell. Second male gamete, the other male gamete combines with the secondary nucleus that is present between secondary or the polar nucleus which combines to form a triploid structure called primary endosperm nucleus. That is PEN. That is abbreviated as PEN. Thus, the two male gametes combine each with an egg cell and a secondary nucleus. The one male gamete that combines with the secondary nucleus results into the formation of endosperm, which is a part of post-fertilization event. Thus, the combination of male gamete and egg cell forms a diploid zygote and embryo, which is a part of post-fertilization event, and that with the secondary nucleus forms endosperm, that is triploid structure, primary endosperm nucleus. Since the fertilization of two male gametes occurs simultaneously, with each egg cell and a secondary nucleus, the process is called double fertilization. Because two types of fertilization occurs within an embryo sac simultaneously. And so the process of fertilization within flowering plant is known as double fertilization. Such a process of double fertilization is the major characteristic of the angiosperm plants. Now let us start with the process of double fertilization in flowering plants. Before understanding the double fertilization process, that is a process which takes place between male reproductive organ and female reproductive organ, that is the male gametophyte and female gametophyte in plants, Let us understand in detail the structure of ovule and the changes that takes place within. As we all know that an ovule, that is a part of female gametophyte, consists of a mother cell or a megaspore, which by undergoing meiotic division produces 4 haploid megaspores. Generally out of these 4, the 3 disintegrates or become disactive and only one remains active in nature. This is the haploid megaspore which further divides, enlarges, containing a large amount of cytoplasm and divides into 8 nucleate cells. That is produces 8 haploid megaspores. These 8 haploid megaspores that is produced gets arranged in the form of 3 plus 2 plus 3. The 3 cells that are located at the opposite end of the embryo wall opening are known as antipodal cells, which itself gets covered individually with a cell wall. The 3 cells which are located towards the embryo wall opening are known as micropylar cells, which contains 2 synergid cells at the opposite end and in between are egg cell containing egg nucleus. The between 2 cell that is placed in the middle portion are known as secondary nucleus, which are binucleate in nature. These cells undergo the process double fertilization, so let us study this process. As stated earlier, a pollen grain is a male gametophyte part which consists of 2 cells, that is, a vegetative cell, tube cell, or a sperm cell which is known as generative cell, that takes part in the process of double fertilization. This pollen grain lands onto the stigma of a flower via pollination and thereby which starts the fertilization process, or which is the beginning of the fertilization process. Within this, the sperm cell, that is the generative cell, enlarges into a tube-like structure known as pollen tube, which contains the male gametes within. It enters into the ovule and reaches at the micropylar end, which connects with the synergid cells, that is, these male gametes within the pollen tube enters the micropylar end and reaches to the egg cell. These 2 male gametes occur in such a manner that each male gamete fuses with the egg cell and the other fuses with the secondary nuclei, each resulting into the formation of an embryo and an endosperm. Thus, the secondary nucleus that forms a triploid nature is known as endosperm nucleus, while the egg cell one is known as a diploid zygote. In this manner, the hybrids are produced via the fertilization process, which further enters into the formation after going post-fertilization forms the fruit.